Good morning, everybody. Today we're going to be talking about photosynthesis. A good way to remember this is photo. Sounds or represents light. Synthesis means to make. This process is going to use light energy to make sugars and oxygen. So the reactants of photosynthesis are going to be CO2, water, and energy from the sun, and it's going to use that energy to build glucose. It's going to release some water, and it's going to breathe out oxygen. You'll notice that the reactants and products of photosynthesis are essentially the opposite, minus sunlight, of cellular respiration. We say photosynthesis is endergonic because it's absorbing all of that heat, and it's using that energy to anabolize or build up large sugar molecules. So it's going to build sugars. You do need to know what the reactants versus the products are for the process of photosynthesis. This is going to be the foundation of food chains. So even if you only eat meat, a lot of people like the carnivore diet, somewhere along the line, you're eating an animal, and that animal that you ate an animal, and then somewhere along the line, an animal was eating a plant, or an animal was eating an insect that ate a plant. It all comes back to plants. That's where the energy from the sun can be used to make sugars or food for the food chain moving up. So when we look at a food chain, it's all going to come back to plants, and then plants get all of their energy from the sun. We call these primary producers, the first producer in the food chain. And then everything after that is going to be some type of consumer, whether that's primary, secondary, tertiary consumers. If we look at a leaf, the structure of a leaf has been optimized to facilitate the process of photosynthesis, having this nice mesophyll area with a lot of cells that are packed jam full of chloroplast. And those chloroplasts are going to absorb energy from the sun and then use that to produce sugar and oxygen. They're also going to need water, and they're going to have to bring in CO2 from their stomata. Those leaves are also going to have a lot of vascular tissue running through them. You can see them when you look at a leaf and you see those veins running across the leaf. And that's so that nutrients can be brought to the leaf, and then also the sugars and oxygens that are produced can be transported down throughout the plant to feed the plant, because the plant's still going to need to do cellular respiration. The parts of a leaf are going to be important, so we'll go through the different parts of a leaf. The outer covering, we call that the cuticle, which is that waxy coat of the leaf. If you've ever tried to break a leaf that has a strong cuticle, it'll be plasticky. It snaps, and it's very strong. That first layer of cuboidal cells underneath the cuticle, that's going to be, if we're talking about the top of the leaf, that'll be the upper epidermis. And then the bulk of the leaf, and I like to use spinach as an example, the thick bulk of the leaf is going to be called, all of it, is the mesophyll. Now, the mesophyll has two layers. That top layer is more compact, and we call that the palisade mesophyll. And then the part that's more loosely packed is called the spongy mesophyll. And then as we move down the plant, we'll have, well, here you see a collection of xylem and phloem. So that's going to be the vascular bundles. Again, that's for water, food, hormone transport. And then coming down here, we have the lower epidermis. And then we have a cuticle on the bottom side of the leaf. And then here they show you this opening for CO2 and oxygen gas exchange. We call that the stoma. And then the cells that regulate the opening and closing of the stoma, we call those guard cells. And you can remember that by thinking of security guards that guard a door to only allow certain things to go in and out. The guard cells guard the stoma and regulate when it opens and closes. Now, the organelle within those mesophyll cells, you can even see the chloroplast in there. So if we zoom into one of those cells, they're going to have chloroplast in them. And then we can zoom into a chloroplast and see what is the structure that actually performs photosynthesis. What does that structure actually look like? And this is an organelle. It has a few different important parts that we'll talk about. And we'll go ahead and go to this next slide and discuss each individual part. So the chloroplast does have, which is really interesting, it has a double membrane, so 2 phospholipid bilayers next to each other. And the one on the outside is called the outer membrane. This is significant because organelles, unless they were eaten by a cell, when a cell goes through, when it phagocytizes something and eats it, it can wrap it in a phospholipid membrane. And the chloroplast and the mitochondria both have this appearance, which has caused some people to theorize that chloroplasts were phagocytized by an ancestral cell and that these used to be their own bacteria, which is quite interesting. The fluid that fills the chloroplast, they call that the stroma. That's where the light-independent reactions will occur. And then, of course, we have that second membrane that's deeper, and we call that the inner membrane. And then it has all these little flat circular structures that are called thylakoids. And these thylakoids, they look like little solar panels, and that's exactly what they do. They harness photons and use that energy to drive the movement of electrons, just like real solar panels do. And then a stack of thylakoids is called a granum. So again, this is a chloroplast. There'll be several of these in a plant cell, and they'll be used to absorb energy from the sun to drive photosynthesis. Well, what is actually providing the energy for photosynthesis? That's going to be radiation. So we have the electromagnetic spectrum, the variations, and the spectrum of values that we can have for different types of radiation. You'll see gamma rays here, then X-rays, UV radiation, and then visible light. Now, if a wave, you see this wave here is really compressed and then it drags out more and more and more. So what it's showing you there is the wavelength. As the wavelength decreases, right, the distance between the waves gets less and less as we move towards gamma rays. As the wavelength decreases, the energy increases. So low wavelength waves have high energy and they're more dangerous. So as we get down here to gamma rays, we're going to have a lot of energy. We're going to have a low wavelength, and that's going to make them very dangerous. So think about radioactive sources like an atomic bomb, fallout from an atomic bomb. It's extremely dangerous because it's high energy. Whereas we go to the other side of the spectrum, microwave oven, at one time people were very skeptical of using microwaves because they were scared of them. They thought it would cause cancer and create all these issues for people. And just theoretically, just looking at it, the wavelength is so great that the energy is much lower. Now you can concentrate it, and like a microwave does, and it heats up food. But if that's escaping the microwave, it's not going to harm you because it's lower energy than just the light coming out of the light bulbs in the kitchen. But if it's concentrated in one place, it can heat things up just by creating friction and shaking the molecules around. So anything lower than visible light is going to be safe in low doses, and anything higher can start to become dangerous. Like UV radiation comes from the sun, and that can cause cancer, and it can mutate your DNA. Now, what's going to be used to drive photosynthesis? We'll talk about the visible light that our eyes are designed to see coming from the sun. White light coming from the sun has all of these colors in it. And we'll look at which colors are being used to drive photosynthesis. Sunlight is also going to have that UV radiation and infrared radiation. And you do need to know the wavelengths of light And a way to remember that, the different wavelengths and colors, is Roy G. Biv, red, orange, yellow, green, blue, indigo, violet. So all of this white light comes from the sun. Some of it is absorbed by certain surfaces. Some of it is reflected. When a color is visible, what that means is when you look at an apple, all of the light from the sun is absorbed, and only certain colors are reflected. In this case, red is being reflected off of the apple. So red is what's going to come to your eye, and the apple's going to look red. So a color that is visible is a color that is reflected. A color that is not visible is a color that is absorbed, right? Purple, blue, green, black, all of that light is being absorbed. Therefore, you're not going to see it because it's not being reflected. So when we look at plants, plants look green. The reason they look green is because green light coming from the sun is not being absorbed by the leaves. It's not being absorbed by chlorophyll. So what that tells us, when you see a plant and it looks green, that means it's not using green light to power photosynthesis. Because if it was, it would be absorbed and we would not be able to see it. Plants have other types of plants other than just chlorophyll. Chlorophyll reflects green, but you also have carotenes. These reflect yellows, oranges, reds. You'll see these in carrots. And then anthocyanins, these reflect purples. You might see that in an eggplant. So based on the color that you see, that's the color that was reflected off of the plant. And these different pigments give us different colors. So now we're going to talk about photosynthesis. Here is just a big summary of the whole process. And you really want to make sure you understand just overall what's going on before you get into all the little details, because there's a lot of details. And the details don't matter if you don't understand the big picture. So let's talk about the big picture. So again, this whole thing here, this large organelle, this is a chloroplast. So all of this entire process is taking place in a single chloroplast. Now there are two processes in photosynthesis that are happening. One of them is the light reactions. We see that on the left. The other is the dark reactions, or it might be called the light independent reactions, or I'll just call it the Calvin cycle. The light reactions occur on the thylakoids, those solar panels. The Calvin cycle occurs in the stroma. Each process has different ingredients and different products, and you can see how they're interrelated. The light reactions absorb water and sunlight. The thylakoid will absorb the sunlight and use that to split water in a process called photolysis. And that water, the hydrogens and oxygens and electrons, will be used. Oxygen will be a waste product, so it will come out. But hydrogen and the electrons will be used to make ATP, NADPH. And the ATP and NADPH will be used in the Calvin cycle along with carbon dioxide, because plants have to breathe in carbon dioxide, to make sugar. ATP and NADPH, they're not just consumed, they're used like a battery. And then once they've been used, the adenosine triphosphate becomes adenosine diphosphate, in which case it needs to go back to the light reactions of the thylakoid to be recharged with another phosphate. Same thing with NADPH. After it's been used for the reduction step of the Calvin cycle, it will become NADP+, and it will have to go back to the light reactions to be recharged. So ATP and NADPH just keep going back and forth and back and forth. They get recharged by the thylakoid, then they go to the Calvin cycle to make sugar, then they get recharged by, and you get the idea. So these two processes are interrelated. The light reactions feed into the Calvin cycle. The ingredients, we see light, water, CO2. These are these intermediate molecules. And we're going to make oxygen and sugar, and a little bit of water is going to be released as well. So the main thing to keep in mind with the light-dependent reactions is the name tells you what it is. It requires light. There's these different photosystems, and interestingly, they have the opposite name that you think they would have. For example, light actually comes in and the process starts on what's called photosystem 2 instead of photosystem 1. The reason that's the case is because photosystem 1 was discovered first, and then they found photosystem 2 later, and they're like, well, we'll call it the second one, but then they learned that the order is actually not the case, that it starts there, but then they just left it the same. So now you have to learn it backwards, but everything gets started right here at photosystem 2. So this is the general process here, and there's a lot of steps to it, and it looks super complicated, but it's actually not that complicated. So we'll just go through the whole thing here and describe what's happening. So first we have this chloroplast, and again, we're talking about light-dependent reactions. These occur on the little thylakoid, the little cookie of the granum. So we're going to zoom in to that. And this is the membrane of the thylakoid. And this is the process that's happening. So you'll notice we have water here at the beginning of the process, and we have light. These are the two big ingredients for the light-dependent reactions. The first thing that's going to happen is that light energy is going to be used to split water. Well, what are we going to split it into? Well, it's made of hydrogen and oxygen, so we're going to split it into oxygen and hydrogen. When that happens, we are also going to remove its electron. Its electron is going to get energized by that light energy. So the light energy zaps the electron, it gets excited, and then the electron wants to move through what's called the photosynthetic electron transport chain. We'll see a very similar electron transport chain in cellular respiration. So the electron gets energized and it starts moving across these proteins. And the proteins, when the electron is inside of them and it's moving through the protein, it's going to make those proteins have a partial negative charge. Remember when we split water, we already got hydrogens on this inner thylakoid lumen, in this inner membrane, or this space inside the membrane. So we already have hydrogen there. As these electrons move through these proteins, they make them negative. Well, that's going to attract more protons on the outside of the thylakoid, in the stroma. So what that's going to do is that's going to pull these hydrogens, because they're attracted to that negative charge, into the thylakoid lumen. So we're splitting water and we're getting hydrogen. And now we're pulling in even more hydrogens. And this continues to happen. And what will occur is the thylakoid lumen will literally swell up like a balloon. It starts to swell and fill with all this proton pressure. Well, we're not done yet. The electrons continue to get energized. Now they're at photosystem 1, and then they continue to move. The electron's not going to be wasted. There is nothing in the cell that is pointless. It's all purposeful. And this electron's not going to go to waste either. So what's going to happen is, because we have to keep the electrons moving to keep that difference in electronegativity to continue, so that we can continue to split water and keep the process going. So as the electrons move, they continue to get energized. Then we're going to use that electron to reduce, reduction is gain of electron, an NADP+. When we add an electron to it, it will now allow it to be attracted to a positive proton, and they can slam together to make NADP+. So the electron will be used to reduce NADP+ into NADPH. That's the first thing. Remember, we already said we split water and oxygen is being released. So now oxygen is coming off, NADPH is coming off. The ingredients were light and H2O. Well, now we've got this huge lumen full of protons. So what are we going to do with that? Well, the cell makes one opening that the protons can go through, and it just so happens that that opening is called ATP synthase, and ATP synthase uses the protons that slide through it like a water wheel. It's going to start spinning and spinning and spinning and spinning and spinning. And as protons pour through, it spins faster and faster, and it takes phosphate and adenosine diphosphate, slams them together, and makes adenosine triphosphate. And then the protons go back into the stroma so that the process can continue. Again, protons can be pulled in through this whole process. And it seems like a lot, it's all complicated, but really if you just look at what's coming in, what's coming out, it makes a lot of sense. Light is coming in, water's coming in, oxygen's coming out, NADPH, ATP is coming out. So that is the light-dependent reactions occurring on the thylakoid membrane. Now, what's actually channeling those photons is these chlorophyll pigments on top of photosystem 1 and photosystem 2. The photons get energized and they bounce from chlorophyll to chlorophyll, and they get to the reaction center chlorophyll, which then allows it to be maximally energized, and it will jump and be ejected to wherever it has to go next, depending on where we are in the process. But it's going to be carried from protein to protein so that electrical energy can be used to drive the proton gradient so that we can make ATP later. And then eventually the electron will hop off and be used to make an NADPH. So again, backing out here, zooming out, we just zoomed into a little segment of a thylakoid in a granum in a chloroplast. So remember, we looked at water came in and was split, sunlight energized those electrons, oxygen came out, and then we looked at how NADPH and ATP were used, or produced, sorry. Now we're going to look at how NADPH, ATP, and CO2 will be used to make sugar. So we call these processes or reactions the light-independent reactions because they do not require light at all. And they do not occur in a thylakoid. They actually occur in the stroma, in that fluid filling the space where all those protons were. So you'll hear it called many different things. You might hear it called Calvin cycle, light independent reactions, or the dark reactions. I'll mostly call it the Calvin cycle. It's just what I'm used to. So, wow, that's a lot. So we're going to split it into these 4 steps. And again, think about what's happening. We're using CO2, ATP, and NADPH to make sugar. So what are all those things doing? Well, primarily, CO2 is being brought into the plant in those stomata. CO2 is a carbon-containing molecule. Carbon is a great resource for building biomolecules, as we learned in the macromolecules lecture. So CO2 can come into the stroma of a chloroplast, and if we have high potential energy molecules, we can use those to drive the modifications of carbon dioxide and make it whatever we want. We just got to have potential energy molecules to modify it in the way that the cell needs it to be to produce sugar. So carbon fixation is bringing that carbon in initially. The reduction phase is using these molecules that we produced in the light-dependent reactions to modify that carbon-containing molecule as much as we can so that we can turn it into sugar. So reduction will be taking place. Reduction is gain of electrons. That's what the ATP is. They're split. In NADPH, we donating their electrons and their hydrogens, and they'll be using that energy to modify the structure of this carbon-containing molecule. And it will happen over and over until eventually we get what's called G3P, which is half of a sugar or half of a carb. It's half of a glucose. So the third step is making those G3P molecules. And that's what we call carbohydrate formation. And then what happens is we have to recharge the protein that facilitated this whole process, which I always called Ruby, RUBP, ribulose bisphosphate. And the way to imagine Ruby is what I've always called it, is it's like a dump truck that is driving around. It picks up the CO2, drives it around, gets modified. Other things are coming into the dump truck, mixing, and then it dumps its container, which is half of a sugar, and then it comes back up, gets more carbon dioxide, goes back down, gets modified, dumps its container, comes back up. So we call that dump truck getting refilled Ruby regeneration, or ribulose bisphosphate regeneration. So again, CO2 is coming in, it's getting reduced, it's getting modified to half of a sugar molecule. Then those molecules are being released and combined to form glucose. Sometimes it'll be broken down into 3 steps, and they're just showing you that sugar formation, they're just not calling it a step, so it depends on how you classify it. But you'll see that the products from the light-dependent reactions are the ingredients for the light-independent reactions. NADPH and ATP are being used here to modify these molecules, and CO2 is coming in as well, which is why plants need to breathe in CO2 to be able to do photosynthesis. Well, how do plants get the water in the first place? Well, they've got to take it in from their roots, and the stoma allow them to create a vacuum to pull water from root to shoot. So when they open their stomata, water and gases can leave, and just like a siphon, it creates a negative pressure up here and a positive pressure in the soil that drives the flow of water naturally. And due to cohesion and adhesion, as we talked about in previous part of the semester, you'll know that water wants to stick to itself and stick to the tree, so it will naturally climb the tree with a difference in pressure. CO2 is going to be entering through the stomata, and O2 is the waste product from the light-dependent reactions of photosynthesis, so it will be released. So those guard cells are going to regulate when the stomata open and when they close. Some plants, they can modify when they open and close their stomata based on the environmental conditions that they're in. You know, if it's really dry and hot and windy, a plant in that type of climate does not need to open its stomata during the day. And why is that the case? Well, if it did, it would be so hot, all of its water would pour out immediately, and it would shrivel up and die. So plants have modifications to change when they open and close their stomata, and even where they do certain parts of photosynthesis to preserve water in their tissues. We'll see this with corn and cacti. Two of these other pathways are called C4 pathway and the CAM pathway. The C4 pathway, what the plant can do, because it can't open its stomata to bring in CO2 because it's too hot, we see this with corn, they just make their own CO2. And the issue with that is it's really expensive, but it allows them to survive and not become dehydrated. The CAM pathway is what we see more with cactus, and they open their stomata at night. So they change when they open their stomata. And that is all for this lesson.